So what I'd like to do is give you a little bit of an overview of my career in research, and also a little bit about UQ-CCR, and then some sort of general philosophical comments. So first of all, I'm pretty ancient. I started at UQ in 1983, and this was a time when people went straight from high school into medical school. It was also a time when we just had the Commonwealth Games in Brisbane, and the big mascot was Matilda. And then the Regatta Hotel was sort of the main university hotel, and the whole Coronation Drive was blocked off one day when I was in first year, because Australia won a yacht race called the America's Cup, which is a bit bizarre to think that would be so important. So what we did is we spent the first 3 years at St Lucia, and then we went to Hurston. And it's quite interesting when you look at the medical school building at Hurston. They actually call it the medical school. So it's not University of Queensland Medical School, it's the medical school. So I always think that's pretty good. So I'm sorry, this is a bit yucky, but I got into infectious diseases because I was quite interested in tropical medicine. And one of the sort of historical things about research at Brisbane was that the condition that causes elephantiasis was actually discovered in Brisbane in the 1800s. And so there used to be people with those great big swollen legs that happens with elephantiasis. But one complication of it as well is that in the old days in Brisbane, there used to be men with wheelbarrows who would push their testicles around because that was the consequence of their elephantiasis, a really dreadful disease. And even now in South Pacific Islands, where it still occurs, this is something that you see. So I liked sort of tropical diseases. But then one day in medical school, one of our, I guess, doctors who taught us brought in a patient, and there was probably a room of about 20 of us. And he brought in this patient, and the patient had low platelets, and platelets are a part of the blood that stops the blood clotting. And we had to sort of ask questions of the patient and work out what was wrong with them. And none of us could work it out. And I think this doctor thought we were all pretty useless. And eventually he asked the patient what was wrong with them. And the patient said that he had AIDS. And you have to remember that in the 1980s, and actually until 1996, basically everyone who got AIDS died. And, you know, that as a young medical student who was sort of interested in research, that really, you know, when you've got such a topical hot issue with 100% mortality, you know, that was really something very interested in potentially researching. And so this doctor was actually Ian Fraser. So in the 1980s, he worked very much as a doctor. I didn't realize he was doing HPV research, but he's an immunologist. And I guess that ties in with vaccines, but it also tied in at that time with HIV, because HIV attacks the immune system, and particularly the CD4 cells. So after finishing medical school and becoming a consultant doctor, I enrolled in a PhD through UQ. And at those times, you could actually spend a little bit of time at UQ, but you could do most of your research overseas. And so I went to the US, and I went to this city called Pittsburgh. And I did things that I wouldn't recommend to you in that my research was actually on antibiotic resistance. But when I first got there, I was really excited because by this time it was 1997, and an effective treatment of HIV was available. So I did an HIV clinic, and I was at a conference, and somehow I was just lining up for a coffee, and I was talking to this American guy, and he said he had this technology where he had a little computer chip in the sort of lid of a pill bottle. And basically what you could do is work out the date and the time that the pill bottle was opened, and presumably that you took your medication. And so I thought, wow, that's quite neat. And what I did is in my HIV clinic, we got an ethics approved consent that we could measure the patient's adherence with their anti-HIV medications. And that actually we found was very tightly linked to whether they lived or died. In fact, you had to take more than 95% of your pills to enter into the highest survival bracket. And at that stage, it was interesting because normally the sort of mantra was you only had to take 80% of your pills for them to have an effect. So for example, in blood pressure, you only had to take 80% of your pills and you'd have adequate blood pressure control. But we found that the bar was much, much higher with HIV. And so I studied, I sort of published that, and because it was a highly topical area, and because it did have a lot of impact on people, it was a very widely cited article. And so I guess the first thing I want to give to you is, take advantage of opportunities. If there's something topical that you're also quite passionate about, that's going to drive you in terms of your motivation to study it, and is also potentially going to allow you to have significant impact. And, it was so weird. About 10 years later, I went to South Africa, and I found out that every hospital pharmacy employed a person whose job was to count the pills of people who had HIV and to see whether they reached this 95% adherence threshold. And that was so bizarre that my research, which was just done in this clinic in Pittsburgh, had global implications in that people in South Africa were, on a routine basis applying that. So as I continued to work in Pittsburgh, HIV became very much controlled. And what I saw there, because I did a lot of clinical work in ICUs, is that people who were very sick sometimes got infections that were resistant to every single antibiotic. And this is a road in Bolivia called the Road of Death. And treating these patients was like driving along the road of death. If you ended up by bad luck having an infection that was resistant to every antibiotic, it was like another big truck coming towards you from the opposite direction on the road of death. You didn't have many opportunities for escape. And unfortunately, that was what we found, that more than 80% of people who were critically ill and developed a multi-drug resistant infection would die. And so my research, even though it was in this general field of infectious diseases changed so that I changed to antibiotic resistance. And I guess as an Australian, that had some significance because, I think we're the only country in the world where on our money was celebrated the discovery and development of antibiotics. This was Florey, who won the Nobel Prize for the development of penicillin, along with Fleming, who was the guy who discovered it. And so even in Florey's time, they found bacteria that were resistant to antibiotics. And one important one was Staph aureus that was resistant, and that was called MRSA. But what I was seeing in Pittsburgh was actually gram-negative bacteria like Pseudomonas, and that was having a really big impact. And so I shifted my study to study of these gram-negative bacteria. Now, it was really weird. The University of Pittsburgh opened a hospital in Italy. And so my wife and I and my two children went to Sicily to work. And that's one of the amazing things about medicine, but also science, is that it gives you the opportunity to live and work in so many different parts of the world. And so, studied and worked in Australia, in the US, and now in Europe. But one interesting thing I learnt when I was in Italy is that just as there was that discovery of penicillin, the Italians actually discovered a whole new class of antibiotics called cephalosporins. And it was as a result of someone reading the literature. So a microbiologist read about this discovery of penicillin, and that was actually from a fungus. So this blob here is actually a fungus called penicillium. And what this microbiologist in Italy did is this was in the sort of 1940s, just after World War II finished. And instead of that water being lovely and clear, it was really murky and brown. And he hypothesized that it was full of E. coli. And that was because raw sewage was just being pumped into it. And he was right. But he also noticed that there were some clear patches within that murky water. And he hypothesized that it was clear because there was no E. coli, and there was no E. coli because there was some sort of antibiotic in there. And so, in fact, what he grew from the clear water was not any bacteria, but a fungus. And this fungus was called Cephalosporium. And so it was like penicillium in that it was a fungus that actually produced antibiotics. So that led to the development of a very, very commonly used class of antibiotics called the cephalosporins. But unfortunately, like all things in infectious diseases, the more you use it, the quicker you lose it. And so bacteria were discovered that were now resistant to cephalosporins. And in fact, that's what I did my PhD on. Infections with beta-lactamase producing organisms, because it was a common problem. It was a problem I saw every day. We didn't know why people developed them. In fact, it was discovered that part of the reason was that a lot of antibiotics are used in food or in agriculture. And then if you eat poorly cooked meat, for example, that meat was contaminated with antibiotic-resistant E. coli, and then that would then populate our gut. So my PhD was a mixture of sort of clinical work, but also molecular work looking at genes that were responsible for this beta-lactamase production. Now it's become a really topical and interesting area subsequently because of this whole impact of what's happening with antibiotic use on our microbiome. And, it really is a wide-reaching area that has reach into many parts of medicine and that, many people have done their PhD on. So I know I don't have a lot of time, but I guess through my career as being a researcher, these are, I guess, some of the lessons that I've learned. So first of all, you've really got to display integrity and honesty. If there's one thing that is going to absolutely sink your career in research, is if people doubt your results, not because they don't agree with them, but because they feel that there's been some sort of fudging of results, rejecting results that don't fit your hypothesis, for example. And I can't emphasize enough the importance of, even if it means your great story doesn't come together properly, you've just got to tell it as it is. And if you don't, your career is going to be completely ruined. The second thing is that no one can do things by themselves. Everything requires a team. And within that team, I'd like to be very positive, but on the other hand, there are times, for example, when your supervisor or your mentor is going to have to say to you, there's an area that needs improvement. And my philosophy is that, everyone can make mistakes, but I really try to work hard to not make the same mistake twice. And if you can avoid, particularly if you've been told something, not doing it a second time, I think that's going to make you look good. No one expects you to know everything or to do everything perfectly, but respond in an appropriate way to any critique of your work. The other thing is that to be really successful as a researcher, I think you need to be very strategic in that if you're doing something for one particular purpose, so for example, you're writing the opening chapter for your thesis, not only is that going to be a chapter for your thesis, but you should get that published. Now, your grandchildren are going to be able to go to PubMed or some other search engine and find the publications that you put out as part of your PhD. And I really do want to emphasize the importance of publications. And in other words, if you're doing something, putting it down on paper, make sure you get it published. The fourth thing is be opportunistic. Just like I showed you, this AIDS epidemic was around, and even though that, I didn't have any great expertise in virology, etc., taking the opportunity to study it, paid some dividends. And in the same manner, show to your supervisors and to others that you're enthusiastic about your work. And that doesn't mean, I acknowledge that many of us are not extroverts, but you should be able to show in your own way your enthusiasm about the work that you're doing, that you're happy to go the extra yard, that it's not just a sort of a 9-to-5 job, and that, sometimes you'll be, on a weekend or other times, suddenly, you might have a burst of enthusiasm and you go and find some papers and think about things. Make sure that people know about it when you do go that extra yard, and show you're enthusiastic for the topic. I think it is important in science that you present your work, and many times that will be actually presenting it verbally, And so I think you do have to take the opportunity to practice public speaking, because it really makes a difference if you're a person who can speak enthusiastically about your topic rather than just be reading a script. And I think that does add impetus and gloss to the research you're doing. Finally, clearly you can't spend all of your time doing research. You've got to have outside activities. You've got to keep your family close, because there are going to be many times when things aren't going to work out as you want, and you're going to have somebody to help you and to bounce things off and to get you back up on your feet. And that's why I think it's so important to really put your family first, and to be able to both give them support, but when you need it, to have them to give you support as well. So these are some of the, I guess, personal lessons that I've had. Just finally, a little bit about UQCCR. We've got 4 research themes: cancer, infectious diseases, neuroscience, and fertility. I won't go into how UQCCR was established, other than to say that part of it was really about philanthropy. So this man called Chuck Feeney was a guy who had $2 billion to his name. And when he was about 60, he said to his wife, "Do you reckon we can live the rest of our lives on $100 million?" And she said, "Yeah." And he said, "Well, I'm going to give away all of the rest of our money before we die." And in fact, he has done that. He's not dead, but he's given away well over $1.5 billion, predominantly for research, including the building that UQCCR is in. So I'm really passionate about research, and I hope that you see this as a great career for you. You know, there is the opportunity to make a huge difference. So, going right back to 1985, when that Scottish doctor, Ian Fraser, had that patient with HIV, to think that, his research was going to allow us to eliminate cervical cancer, which I'll just tell you, from having looked after people with cervical cancer, it is actually a horrible, horrible way to die. I think we're really lucky that we're doing research in the medical area, because it really does allow us to make a difference to people's lives. And that's something that, should really be very motivating to all of us, because we aren't just sort of studying something that, I've got a friend whose son is doing the German politics of the 1700s as his PhD. And I mean, while that might be interesting, for what we're doing our higher degrees in, I think that really is going to make a difference to people. So thank you, Amanda, for inviting me, and I'll look forward to what's coming for the rest of the afternoon. Thank you.